Good morning, ladies and gentlemen. Myself, Dr. P. Samuel, Assistant Professor, Department of Biotechnology of INR Janaki Ammal College, Sivagasi. Through this video tutorial, I am going to talk about an important topic called electron microscope. It is from the paper Biophysics and Instrumentation, which is being offered as an elective paper by the Department of Biotechnology during the fifth semester of undergraduate studies. Right, with these details, let us move on to the topic. What is an electron microscope actually? Right? So, before we move on to the area, I just want to tell you what is a microscope. Microscope is a device used to observe or to visualize tiny objects. Those objects are actually not visible to the naked eyes. With the help of microscope, what we can do is we can observe or we can visualize those tiny objects with the help of this device. See, there are different types of microscopes are there, but above all, simple microscope, compound microscope, phase contrast microscope, even fluorescent microscope, right? Each microscope has its own sort of applications, right? But above all the type of microscopes which I mentioned before, electron microscope is of great importance because of its uniqueness. What is its uniqueness, right? Electron microscopes are able to produce high resolution images. This is a very important feature of an electron microscope, right? It could able to produce high resolution images of the object which is being under examination, right? Second thing, when compared to all other types of microscope, because where we are using light in one form or other form as a source of illumination, whereas in electron microscope we use electron high voltage electrons which are generated from the electron gun. This is one such important feature to be noted in the case of electron microscope, right? So, in electron microscope, high velocity electrons are used as a source of illumination, and this microscope was built by Ernest Ruska in the year 1932. Right, note it in your notebook, because these kind of questions may be asked in quiz component of your paper. Kindly note it down. So, electron microscope was built by Ernest Ruska in the year 1932. Right. So, what is the working principle of electron microscope? Here I want to concentrate more, because as I told you in the previous slide that electrons are generated at high velocity, right? They are produced at high velocity, and these produced electrons, right, are converged, which means they are focused onto the specimen with the help of electromagnetic lens, because in case of normal microscope, to converge the light onto the specimen, we use a component called condenser, which is made up of glass, right? Because in case of electron microscope, the fast moving electrons are quite impossible to be targeted onto the specimen by a simple glass, because those glass condenser will be broken when it is hit by fast moving electrons. To prevent that, here we are using electromagnetic lenses to focus the fast moving electrons onto the specimen, right? And throughout the course of the experiment, the electrons are quite continuously being accelerated. The acceleration rate is ranging from 100 kilovolt to 1000 kilovolt. So this is the average velocity or average acceleration of the electrons which is being generated by the electron gun at the top of the electron microscope, right? So here you have to understand the basic difference between electron microscope and all other microscope. In all other types of microscope, what I have mentioned earlier, like compound microscope, phase contrast microscope, there we are using light as a source of illumination. But in the case of electron microscope, fast moving electrons are being used or being served as an illuminating agent to illuminate the specimen, right? Keep this point as clear, right? Next slide. Imagine this as your object, right? This as your object, right? Maybe a piece of tissue, right? Which is being examined, right? So this tissue is not as such being subjected to examination purpose, because in case of microscopy studies, the specimen has to be very thin. If the density of the specimen increases, we couldn't able to obtain a clear image about the content within the cell. Similarly, in the case of electron microscope, this has to be sliced into small pieces, right? This slice can be done with the help of a device called microtome, right? So using microtome, a slicer, right, a cell slicer, which help us to slice the cells into thin slices, right, to cut the cells into thin slices. This is very, very important, right, because the density as well as the refractive index of the specimen are playing an important role in obtaining clear image, right? So for that, we are preparing a thin slice of the tissue which is going to be studied with the help of a device called microtome, right? Once the tissues are sliced, right, it is mounted on a stage, right? The specimen is mounted on a stage. Fine. After it is mounted, right, after it is mounted, the tungsten filament is switched on so that the electrons are generated, right? Here the source for the production of electron is the tungsten filament. When it is switched on, it started to produce fast moving electrons, right? Here we have to note this point, because as the electrons are coming out of the electron gun in this direction, right, electrons are not moving on a straight plane, right? Their movement is not in a linear path, right? They move in a zigzag pattern, right? Their movement is not like a straight line, right? They will move like this, right? They will move in a zigzag pattern, because this has to be transformed into a straight plane. So how can we do that? This can be achieved by placing electromagnetic lenses on both sides of the electron path, right? These electromagnetic lens will take care of the path of the electrons, which converge the fast moving electrons onto the specimen in a specific pattern, right? So that the electrons are directly focused onto the specimen, right? Here you can observe the pattern, right? These electromagnetic lenses are on both sides, and this will prevent the zigzag pattern of the electron movement. These electromagnetic lens will focus the fast moving electrons onto the specimen in a clear way, right? Once the electron falls on the specimen, some electrons gets scattered, right? Some electrons may penetrate through the specimen. The penetration is an important step because the penetration can be maximized through the slice size of the specimen being taken into account, right? So a laboratory working person is taking utmost care to slice the cell of very thin size in terms of nanometers, because as I told you before, the image, right, the final output of the specimen being examined, which is obtained in the screen, is depending upon the thickness of the specimen, right? When the thickness is lowered or when the thickness is so thin, it is very sure that we could obtain a clear image of this object being examined, right? And the specimen, the penetrated electrons falls on the screen called a projector lens, right? Here I can give it. It falls on the projector lens, and the projector lens will produce the final image. Imagine this as the object, or this is the specimen which is produced by an electron microscope, right? As I told you before, electron microscopes are quite important because of its uniqueness in producing high resolution images, right? This is used in industries as well as this is used in the field of diagnostic microbiology to identify or to characterize the structure and the shape of an organism. Even it is also used in research laboratories to study the ultrastructure of a cell. Only one disadvantage is associated with electron microscope. The thing is, electron microscope is not suitable for observing cells in a living condition. Second thing, the specimen should be free from water, because water simply changes the flow of the experiment, right? So care should be taken while preparing the specimen. It should be completely free from water droplets, right? Thank you so much for watching this tutorial. I am sure this tutorial was quite interesting, and I hope it will give or it will throw some important information in your mind regarding electron microscope. With this I conclude. It's bye-bye from Samuel. We'll meet in another video tutorial. Bye-bye.